Hello, everyone. Welcome to our varvis webinar entitled A Complete Software Solution with IVDR Certificate. So we will be going from raw data processing to variant classification. So first of all, I would like to introduce myself. My name is Jimena Escalera. I have a PhD in biology, and since last year, I'm a business developer at Team Varvis. I would also like to introduce Varvis to you. We are headquartered in Germany, at the north of Germany, right at the Baltic Sea. We are a software development company and a medical device manufacturer. This means that how we produce and operate our devices is is compliant with international standards. The software varvis is cloud-based, is a clinical decision support system, and it has been recently certified as a Class C device under the IVDR regulation. So we are, for those that are not acquainted with varvis, we are leading the German market. So here you can see some of the labs and university clinics that are currently using our software, our medical device. And I want to talk a bit about the structure of this webinar. First, we will have Dr. Ben Lisfield. He will give us a talk about, in general, NGS software solutions with IVDR certificates. And then we will follow to an introduction and a demo. This will be performed by my colleagues, Dr. Yvonne Kasman and Irene Patrick. And then we will have a Q&A session that I will be moderating. In this regard, in the upper right corner of your screen, you have like the symbols with a question mark. There you can type your questions, and we're gonna try to answer in between the talks and also at the end. We also want to do this a bit interactive. So we have prepared some polls, and since we are all immersed in this Eurocup, maybe we can start with a test question to see how this is working. So now you should see a poll in your screen. So maybe you can answer what is your secret ritual to bring good luck to your team. It doesn't necessarily have to be a soccer team. Maybe you're more involved in other sports. Okay. I'm looking at the answer, so I'm gonna give you some time to answer. Okay. It's funny. So I see that only half of the people is voting. So come on, tell us what are your lucky. Okay, so I will end the poll now, and you should see the results. So we don't have many people wearing lucky jerseys, and it's more like this audience is non-rituals, very scientific. Okay, so I will close the poll, and I'm happy to introduce the first speaker, Dr. Ben Lisfield. So Ben has a PhD in physics. Afterwards, he went to a startup for ophthalmological medical devices and development of computer-guided laser surgery devices. Then he went into the genetics field in a clinical laboratory for genetic diagnostics. In there, he established the IT and the software development and bioinformatics department. Afterwards, he became the co-founder and managing director at Limbus Medical Technologies. So, as I mentioned before, he will be speaking about NGS solutions that have been IVDR certified. So the screen is yours, Ben.
Thank you, Jimena, for the kind introduction. Um, well, our team has been dealing with regulatory topics for the past 12 months quite intensively. And we were happy to be able to announce at ESHG in Berlin just a few weeks ago that our software has been certified according to IVDR. And when discussing this with customers, it appeared that not everyone is so familiar with these topics like we are now. And there's certain important information sometimes missing. And this is where the idea actually was created, that it would be good to have a webinar and explain kind of the implications and the benefits of this kind of certification for a software in the genomics world. So I'm not going to touch kind of the basics of in-house tests. We had several presentations and webinars about this topic before. I would like to refer you to our webinars page on our website, to our recordings. I will only touch them briefly. I will rather focus on the significance of now our software being CE labeled or IVDR certified, and how this can help you in your regulatory strategy for your own in-house test. So just as a short overview, varvis has been classified as a Class C device. I'll explain in more detail shortly what these classes mean, but Class C is specifically meant to be applied to genetic testing, IVDs, prenatal testing, cancer diagnostics. So this is where we nicely fit in, where we think our software should be classified. The classes A, B, C, D um, are defined according to risk. So the higher the class from A to D, the higher the risk for the patient, the potential risk for the patient. And class C is obviously the second highest class. So it entails a lot of quite a thorough analysis, in particular of the risk that's posed to the patient. What is exactly the intended use and the scope of the certificate? Because this may vary from device, from manufacturer to manufacturer. If you look at the entire NGS testing process, which is displayed here quite generically, the IVDR certificate for varvis really covers everything from the raw data to the reporting of a variant. And this is kind of a unique a value that we provide at the moment. There's no other software really providing a certified solution for this entire process. The varvis software is agnostic to assays, to the specific enrichment kits. That means you can use any kind of enrichment kit in your laboratory process and analyze the data with our software. So we have performed created a performance evaluation for NGS panels, focused panels, whole exome sequencing, and also whole genome sequencing. The clinical applications that we address with our software are both germline and somatic applications. We support barcoding of NGS reads. So we support, of course, data without a unique molecular identifier, but also single UMIs and duplex UMIs. And the input is also rather general. We support FASTQ files as well as the raw data from the sequencing instrument. Now I understand that this is kind of a new situation, still new situation for laboratories, because they kind of have a dual role. They are both now under IVDR, they're both manufacturer and user of medical devices. And this is kind of a difficult situation. And you probably have to keep also these two worlds a little separate, because it makes your life a little easier if you think in these two terms. So you are a manufacturer and you are user, and the responsibilities are quite different. As the manufacturer of your in-house test, you are responsible for the design, manufacturing, your supply chain, and for the risk analysis, for example, and also for the performance evaluation. But these are steps, these are processes that you perform probably once or several times when you update kind of the design of your in-house test. But this is separate from kind of the execution of the test. That's something that you do in your routine diagnostics. You use the in-house test, you document how it has been used, traceability, who has done what, and accordingly there are different requirements how these roles have to be performed, manufacture and use, and there are different standards you have to follow probably. So typically manufacturers are applying a quality management system according to ISO 13485, while laboratories simply performing tests can use ISO 15189, a standard that's rather focused on the performance of testing and not so much on design and manufacturing. So this is where I would like to ask you the first question. What is, I mean, since there are guidelines now, also from official bodies who recommend that laboratories introduce a quality management system according to 13485. And I would like to know, has any one of you working in a health institution already introduced a quality management system according to 13485? It's a pretty comprehensive standard. It's not an easy task, but I'm just curious, how many have addressed this task, because this certainly requires a capable quality management team. Okay, I can see that responses are coming in, and yeah, so many of you all, obviously have not introduced 13485 or are not involved with that, but I can see that two of you, I'm closing the poll now, two of you have introduced 13485. This is impressive. I mean, of course, we are working according to 13485, but this is quite a task to do that in a laboratory where we know, the focus is actually on the execution and not on design and manufacturing. A short recap, regarding the requirements that are applicable to in-house tests. And you're probably familiar with the fact that you already have to apply the general safety and performance requirements since 2022, since 2 years ago, basically. There are other specific requirements for in-house tests that I'm simply listing here. These are all items in Article 5, Section 5, and general safety and performance requirements, as well as that no transfer to another legal entity and no industrial scale. These are requirements that have been applicable from the start. B, C, E, F, and I have now been applicable since May, since a couple of weeks. And the only requirement that is not applicable yet is item D, the justification for your specific patient group. This is still delayed until May 2028. But apart from that detail, everything else has already been has to be fulfilled. And again, I'm rather curious, and I'm not judging. I just like to know, laboratories are now required to make publicly available a formal declaration of conformity that their in-house tests are compliant with IVDR requirements. And I would like to know from you, have you seen such a declaration of conformity? I mean, it doesn't need to be your own institution. Could be that you have seen it in other institutions. I'm just curious. I only have seen one so far, but maybe I'm not looking in the right places. But since most of you are coming from health institutions, you are probably better informed than I am. Okay, so interesting. Again, there is a large fraction of you have not seen such declarations of conformity. Again, it's okay. It's a fresh, I have to admit, but since 3 or 4 weeks, these declarations of conformity should be available now, and laboratories are required to draw them up. Let's see how many are going to do that and how this is going to develop in the future. We know this entire process, establishing compliance, is a huge effort, in particular for organizations like laboratories who are not used to be manufactured or who are not used to have this manufacturing role. So let's take closer look at kind of the ideal regulatory approval procedure. What is a good strategy that you can follow to establish compliance with IVDR for an in-house test? At the bottom of the slide, I'm referencing a publication from Spitzenberger from 2021. This is a really useful, very specific publication that also provides templates that can be used in a regulatory approval procedure for in-house tests. And this publication is also referenced by documents published by GfH, the German Genetics Association, and AMWF. And I'm assuming that many other national bodies, professional associations are using those recommendations and, and providing information to laboratories how to use them. So um, these recommendations from Spitzenberger basically say you should start at the intended use, define the intended use for your in-house test. And a very nice form sheet is provided that you can, with lots of check marks where you can fill in the information, which really facilitates the process, and become aware of the inherent risk that your in-house device poses to patients. And taking those two combined, you can come up with a classification of your device. I'll explain in a moment how this classification process works. Even though there are no specific requirements in IVDR that in-house tests should be classified, it makes sense to perform this classification because it may actually make your life easier in many aspects. And from this classification, you basically determine to what extent and to what level of detail you need to document how you comply with the general safety and performance requirements. And remember, these general safety and performance requirements are rather comprehensive. They comprise requirements regarding the risk management of your in-house test or performance evaluation, specific requirements regarding design and manufacture, labeling, and instructions for use. And if software is part of your in-house test, there are requirements regarding software and IT security. When you read these general requirements, your first reaction is probably, these are so general, how should I apply them to my specific test? And kind of the recommended way to address this, to demonstrate compliance, is to use harmonized standards. So these are officially listed standards that you can use, and by showing compliance, demonstrating compliance with those standards, who are more specific, you automatically imply compliance with the general safety and performance requirements. And there are, particularly for software, there are quite extensive standards, IEC 62304 or IEC 82304. So these um, 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 concern everything that's related to software development and IT security. Now let's take a closer look at the classification rules, because I mean we recommend that you apply them. The classification rules in the IVDR are structured in such a way that you basically start with the highest risk level and move downwards to lower risk levels and see in which kind of if you would fall under any of those categories or classifications. And it basically starts with rule number 1, which addresses blood screening, high-risk disease testing. So these are devices that would concern a large number of people, the entire population. These should be classified as Class D. If your in-house test does not fall under Rule 1, you move on to Rule 2. Does it have anything to do with blood tissue compatibility testing? That would be considered Class C. If this rule doesn't apply to your device, you move on to rule number 3, and this is kind of the interesting rule for all of us. It concerns infectious diseases, genetic tests, prenatal testing, cancer diagnostics. All this is under rule number 3. These devices should all be considered Class C devices. If that would not apply, there's another rule about self-testing, would also be Class C. And then if none of these rules 1 to 4 apply to your device, maybe rule number 5 can apply, which concerns very general reagents or instruments used in the laboratory or sample receptacles. Um, and this rule is very interesting because devices that match this rule can be classified as class A, which is the lowest risk class and which very much facilitates the documentation and the compliance with IVDR. Then in the end, if none of those rules apply, there's a catch-all rule, number 6, and this allows you to classify your device as class B, but all devices in all risk classes except class A have to undergo conformity assessment by a notified body. Your in-house devices are exempt from that, but just to give you a context, in general, the IVDR only sees class A as low risk and with lesser restrictions and lesser requirements. This is kind of a simplification of the entire rule set. I've left out a rule, but these are the rules that are interesting for us. So why am I talking about IVDR classification rules? When you are looking at your own in-house test, and this is basically a very generic chain of process steps in, for example, an NGS genetic test, you may be able to identify certain modules that you could isolate, that you could treat as individual devices. Let's take, for example, DNA extraction and the sequencing. The DNA extraction is even listed or mentioned in a guidance document specifically. It's a very generic process step, and you're probably using it in many different genetic tests. So it's the same process or the same components that you're using in different tests. It would make sense to identify this as its own independent small device. You can classify it internally as class A and have a very nice compact documentation how you fulfill the general safety and performance requirements. On the other hand, also sequencing devices have been mentioned in guidance documents. It's possible to also label them as class A. Again, facilitates the entire process. There may even be commercially available devices in the near future, or it would simplify your own qualification that you have to do with if you're using research use only devices. Now let's take a look at another process step or a sequence of steps, library preparation. I just summarize it here as one rectangle, but it may comprise, of course, many different components and steps to be performed. And when you look at this in more detail, you may find out, one part of it are rather general reagents that would fall, for example, under Rule 5. But there may be very specific, test-specific components. And even if those components are available as CE labeled devices under IVDR, we've seen such components be classified as Class A, which inherently means the manufacturer did not identify any inherent risk. But if you yourself as in-house test manufacturer, if you say, well, there are components who are critical, if they fail or if they are not designed properly, this has grave impact on the diagnosis. Let's take, for example, you could, for example, consider the barcodes. When you're multiplexing samples, the barcodes, you need to be sure that those are manufactured correctly, or oligonucleotides that you use for enrichment. We all know that lots of things can go wrong in the enrichment process, which has impact on the diagnosis in the end. So you may disagree, or your risk assessment may differ, and then you have to pick it apart and treat the components in different ways. But in general, I would think, or from our point of view, it is helpful to think in a modular way and address and also perform the risk assessment very specifically. Now let's take a look at software, which is rather at the end or the second half of the process. Let's assume you are using your own bioinformatics pipeline or a commercial research use only software or some open source software from a third party. If you want to integrate this in your in-house test, you have to demonstrate that the software has been developed according to the state of the art. This is a very broad term. It comprises lots of requirements. State of the art means it needs to follow the most current international standards, for example, which concern the life cycle of software. So the standards that I mentioned before. And this may also comprise, for example, security testing, penetration testing, if your software has some connection to the internet. Again, this would be, if you're using a piece of software like this can be a lot of work. We know this because we basically have gone through all this. And you may, for the second part of the software analysis, you may be able to use an CE labeled device for the filtering and reporting of variants. We've also seen that that these devices have been classified as Class A, something that we cannot really follow when looking at kind of the classification steps that I showed to you earlier. It's really about, you really need to take a close look at the intended use. What is the manufacturer saying, what it is meant to be used for? And again, kind of the classification tells you what is the assessment of the manufacturer, what the inherent risk for the patient is. If you disagree with that, you're basically using the device in an off-label kind of way, and you have to make sure that your risk management is sound and complete, and which would mean additional work for you if the classification does not match your own assessment. Now let's take a look at the varvis software with its Class C certification now. The varvis software covers all software steps in this process. It starts from the raw data. It's one uniform device, and hopefully it addresses all the use cases that you have regarding your NGS in-house test. And the benefit of the varvis software is it's clearly um, classified as Class C. So we already classified it in such a way that we consider it um, as a critical component. The way that the software works certainly influences the outcome of your diagnostic test. Uh, we um, we created a very extensive performance evaluation that has been reviewed in detail by a notified body. We performed risk management, and we are continuously monitoring IT security and had external penetration testing. And this makes life very much easier for you when documenting your in-house test. You basically don't have to concern yourself with the software part anymore. You can simply refer to the Class C certificate that we can provide to you. And this in particular means you don't need to validate any further that the software works. We are certifying that it works. Actually, in addition to that, we offer even more useful services to you. So, for example, we provide you with a platform test report as required by international guidelines. So you can sequence reference material that has been very well characterized, for example, genome in a bottle reference material, and we provide you with an automated and standardized report about the analytical performance of your platform. The platform meaning both your wet lab part and our bioinformatics analysis part. And what is more, now that we have the IVDR certificate, we can even offer you a simplified platform test for the CNV analysis. So this saves certainly a lot of time and investment into validation procedures. But kind of the utility of varvis goes even further. Remember, you have the dual role. You are manufacturer and your user. As a manufacturer, you need to maintain risk control. So you may identify, for example, in your wet lab process, certain steps that can go wrong, and you need measures. You need to document measures. How can you minimize that risk? How can you identify that something has gone wrong? This is there are features in varvis that allow you to do that, because many errors that occur in the wet lab process can only be determined or can only be recognized in software. So we provide you with a wealth of quality metrics like demultiplexing report, for example, any errors with index barcodes can be identified there. We have examples for that. We provide information about coverage, about the hybridization efficiency, which allows you to judge have these steps in the wet lab workflow performed well. And we even provide you with metrics, with scores that allow you to identify mix-up of individual samples or contamination with a foreign sample that's not even in your NGS pool. And we verify, for example, also the sex of the sample from the NGS data. And all this can then also be used for your post-market surveillance. All this information is stored in the varvis database and allows you to continuously monitor the performance of your in-house test. And all these are your responsibilities as a manufacturer, and it's actually extremely difficult to do that without a validated software tool, and varvis nicely provides you with these features. So now, as a summary, kind of the most important messages that I would like you to remember when you leave this webinar is, when you're looking at your in-house test, always start with the intended use and the inherent risk of your in-house test. And if possible, that's a recommendation that you don't need to follow, but it may make your life easier, modularize your in-house test. And well, of course, use CE devices in those modules if possible, particular software. I can only recommend that because we've already done all the work for that. And well, if you're not sure about the CE labeling, check the intended use of your components. Is the component really meant for what you're using it for? If this does not match, you take over the responsibility of the manufacturer, and check if the component classification matches your own assessment. In the end, you're responsible for the entire in-house test, and it's up to you. It's your risk assessment. And you have to make sure that all your components match your own requirements. Okay, so this is kind of a brief overview of how you can utilize our certified software in your in-house test. Of course, I mentioned before, we already have resources about kind of the context, about kind of the general requirements of IVDR on clinical laboratories. We also have blog articles. I invite you to visit our website because many of those webinars have been recorded, and you can review the information that we provide there. Okay, now I'm handing over to the next speaker.
So beforehand, we have some questions, but before I start reading this, I would like to remind you that in the upper right corner, we have a question mark symbol, and if you press there, you open like a sort of chat in which you can type your questions. So we have maybe one question now, which is, why can manufacturers choose a device classification that does not make sense?
Yeah, a good question. And I'm not sure if I have I have an answer for all classifications. I mean, the IVDR is structured like that it's the responsibility of the manufacturer to determine the classification. And if there's no involvement of an authority or a notified body, that's as it stands until somebody questions that. Again, if you think a device classification is not correct or seems strange, take a look at the intended use, because this is kind of the piece of information that's the basis for the classification, and get into an exchange with the manufacturer what that means, and discuss these differences that you may determine.
Okay. We have another question. Can we use the varvis software as a tool for the validation of gene disease validity?
Oh, this is this is a complex question, I guess. So you want to so this implies that you want to validate the relationship between gene and disease. I mean, kind of I would think yes, because um, first of all, you can utilize varvis, the varvis software, as a validated tool. So you can rely that the functions that varvis provides have been validated by us as a medical device manufacturer. So this is for kind of the first step. It's a validated tool. And that now you have to look at, does the software provide the features that you need to establish this? I can imagine that there are certain features that help you with that, because we store all the variants that you ever found in all your patients. We provide you with the HPO database, and you can certainly utilize your own history of data to demonstrate certain relationships and to gather evidence for certain relationships. But uh, to be honest, can become so complex, or this can be quite a complex project. I would recommend that you get in touch with us, what you really intend to do, and maybe we have done something similar in the past, or we have some ideas how this can be done.
Fantastic. Thank you very much, Ben. And we will continue with the next speakers, which are Dr. Yvonne Kasman and Irene Patrick. They are part of the marketing and sales team, and therefore they are involved in the customer onboarding and in the training. So Yvonne will give us an introduction to the varvis software, and afterwards, or in between, Irene will show us or give us a demo. So thank you very much, Yvonne.
Yeah, thank you, Jimena, for the introduction. Let me see if I can take over the slides. Yes, that works. Great. So, after this kind of theoretical first part, like about your workflows and the validation, we would like to show you now how this would look like in varvis when you work with it in a routine setup. So this arrow we've seen before, and as Ben mentioned, this whole second part is now IVDR certified Class C medical device. As already mentioned before, also we start very early in the process, so we take over when the sequencer is done and the raw data is available. And the consequence here is that not only the variant data is stored in varvis, but you're collecting data on a much broader levels, right? You can see what kind of enrichment kit has been used, which samples have been processed in the same batch. You can review the QC data, the configuration that have been used, the versioning. You can create family relations in the software and review the history. You can classify variants and describe the phenotype of the patients, and then also store the diagnosis. So you, so the level data is stored on all kind of different levels, and varvis itself basically consists of three parts. So the center piece is where you as a user would work with the software, where you interpret the results, you review the variants, there you follow a streamlined workflow. You can classify, prioritize variants using many different features, for example, virtual panels. You can use your invariant and CNV database. You can utilize the inheritance patterns and classify according to ACMG. This we will show you in the software later. Irene will show this. And then we have two components that feed in the data into the varvis software. On the left side, the bioinformatics pipeline, which is an automated, efficient pipeline where CNV and SNV and inter calling is performed. The validation of this is included, as Ben has already mentioned before, and this is also where the QC reporting is generated. And on the right-hand side, you see the annotation management is also done in a centralized way, so that we update regularly the annotation sources that you can use to review your variants, like, for example, ClinVar annotations or Nomad. This is also, you can update previously processed cases with new information. and you can compare your data across the platforms. Before we jump in the varvis itself, I would like to mention that this is basically used for all clinical NGS applications. From targeted panels, we support all sizes, also custom designs, up to like clinical exomes, whole exomes, and also whole genomes. And yeah, also cancer diagnostics, so it's not restricted to germline. Also somatic analysis is possible, and carrier screening. So just to get a feeling about who's actually in the audience, we would like to know what your use cases are. And for this, I have prepared a poll. Let's see. So multiple answers are possible. And please let us know, are you working mostly with targeted panels, so in the germline world, exomes, even genomes maybe already? Do you also look at somatic analysis, cancer diagnostics, or even carrier screening? Gives 10 more seconds. Okay, let's see. Not sure if you can already see the results. I can already see them. End this now. And yes, most of you, more than 80%, are really focusing on exomes. And then we have genomes and more distribution of everything else, targeted panels, with exomes clearly in the deeds. And this is also what we see across our customers, that this is the most commonly used analysis form. And this is also the example we would like to show you in the software demo. We'll close this now. Okay, let's take a look at the standardized NGS workflow that you will follow when using varvis. So the data generated by the sequencer can be uploaded to the varvis platform. This can be FASTQ files or BCL files. Then we would perform the bioinformatic analysis, so CNV and SNV. As I said, germline or somatic. The next step would be to connect this analysis data to the patient and family information. You can perform quality control. SNV and CNV filtering variants that are of interest can be classified according to ACMG, can then be reported, and reported results feed directly back into the database together with external annotations and literature information. This information can then be used to filter the following cases and the following analysis. And if you use varvis, the first part is basically done on our end. So this is something you don't need to worry about. So the first thing that you as a user are working with is like connecting the analysis information to the patient information and describing the patient in detail. So there are several options that you have here to describe the patient. In the person edit window, this is a screenshot from the varvis software. We have like 3 tabs on top. And the first one is to, is about person details. Here you can enter details like, for example, the sex of the patient. The second tab is about the clinical information, and the last tab is the family relation, where you can build up trios or more complex family structures. In the clinical information tab, we have the HPO manager, and on the left side, you can browse the HPO hierarchy, enter terms of interest that just to describe your patient, and then by click, select these terms to describe the patient. So on the right side, you would then see the terms that have been selected already. Additional information regarding parentage, for example, can be entered, clinical modifiers, and this information that is entered here can then be also used to I'm going in the wrong direction, sorry. There we are again, can then be used to prioritize the variants in the software. The next step would then be quality control. This can be done on the case level, but also run specific, so that you can review the quality metrics of all some samples that have been processed within the same run. Then again, the CNV and SNV filtering, classification reporting, and this part of the software, of the workflow, we'd like to show you now in the software. And for this, I would like to hand over to Irene.
Thank you, Yvonne. So I will take you through the software. And for this, just give me a second. Okay, so once the samples are processed via our bioinformatics pipeline, the samples arrive here in the tasks view. And from this view, you're able to assign the cases to the colleagues, and you can see all the different information regarding the samples, like the enrichment kits and the clinical information if you've already imported it via the API, but you're also able to enter this information manually. Here in the person's view are all the closed cases that you can go through again, and here you'll also see all the reported variants. Now, in the classification alerts, in case there's a change in classification for variants, you would badge here that indicates the number of variants that have changed in classification. And here you can see the previous value and also value of all the variants. And this is an example of the filter that I have created, and which says show me all the variants with an updated value of either pathogenic or likely pathogenic. And so from here, I'm able to also enter the case and review the variant information and decide what to do. This is the inbuilt QC module where I'm able to go through all the runs, and I can click and open every run to see all the samples within each run. And if there is a sample that has failed a certain threshold, that is also flagged here. But almost for every metric, all the number of samples failing every metric is also indicated here. If we would like to go see the other details about this, you're also able to approve and reject a whole run. But if you've only approved and rejected certain samples from the run, if you mouse over here, you're able to see how many samples were approved and rejected. As the next step, I would like to show you about the virtual panels. We are able to create the virtual panels quite easily by just dragging and dropping a CSV file, but also these gene symbols can be entered manually to create these panels. And here, like you see, there can be ACMG gene lists prepared, and this can be made available across cases. So let's go into the users. So we do not restrict the number of users on the platform. So you can add any number of users, give them different roles so that they have different capabilities to use the software. Now let's go into one case that I have prepared to show you take you through the filtering steps. And this is a trio where the index is affected, the parents are unaffected, and for this analysis we have the SNV and CNV data, but I will only show you the SNV details in this demo. And the enrichment kit information is here, twist core exome. And to go through the clinical information, we can look at it here. It's basically a neuro case with also seizures as one of the features, and it's a male sample. So if you see the total number of variants, it is about 65,000 variants. And I can choose to apply a virtual panel which will fit this case. And because I saw seizures as one of the features, I would like to apply an epilepsy panel, but also there were other features. So I would just choose a large panel with more than 800 genes and apply it. And if you see here, there is a big change in the number of variants that we will see. And now I can choose to apply even more filters at this step itself. Let's say something like 1% frequency, but retain it if the variant is pathogenic. So when I apply this filter, I would see that the number is reducing further, and then the case will load faster. Uh, and now I have to open and review only 192 variants. But like I said earlier, this could be done in different steps, and this is only a first step, but you could also choose to open the whole case with 65,000 variants without applying any additional filters or panels. Let's go to the variant view. Now, in the variant view, what you see here are filter buttons that I have already configured, but the user is able to easily create these buttons from all the information in the columns. Now, the columns have also been configured in such a way to display minimal information, but you can also choose to display any number of columns in this view and make them visible if you'd like. Right hand side, you see the inheritance filters with an intelligent on and off system, and it gets activated based on the information in the case. Now, if we'd like to start filtering the case, the first step would be to say, I would like to look at all the pathogenic, known reported pathogenic variants. And now for this, there is a button which says, show me all the variants that have been reported pathogenic in ClinVar, the local database, and the Alexis database. So I apply this, and then I also can restrict it to variants only in the index. And now I can go through the 5 variants, but I would like to sort them according to the HPO similarity score, which prioritizes variants based on the HPO terms entered for the case compared to the HPO terms of the genes. So I just have to sort it, and then I see that the most relevant variants would come on top. Now, as you can see here, the first 2 are my variants of interest, and they are compound heterozygous, one inherited from the mother and one from the father. So right here, you can see the segregation, and yeah, in case, if this was a case of, let's say, compound heterozygosity, one SNV, one CNV, you would also see this here. And now I can choose to just highlight these variants and not look into them further and come back to them later. But like I said, this is only the first step, but every step from my workflow, I will be able to save it to a report basket, such that I keep a log of all the steps. So when I send it, I can save all the grid information, all the filters that I applied to the report basket, and then this will be made available as an attachment in the report. So this will contain all the grid information, all the filters that I applied, and it will be saved such that I can review it at a later time point. Say after 2 years, I come back to the case, I will be able to see all this information. Now let's look at the case as a regular one that I've not found the variant. I would apply all the other filters, like say, I do not want to see variants that are benign, likely benign, reported in ClinVar. So I would just apply this filter. Then I would like to see variants that have a high and moderate impact. And then those variants that have a stringent, low frequency. Remove variants that are highly present in home and hemizygous states in nomad. And also, as a first step, I can say, show me only genes with known modes of inheritance. And now, together with all this, I can start applying the inheritance filters. And the first de novo filter, I would give a very stringent frequency filter. And now I already see that the one variant that was there is not relevant anymore. And then I can go to the next filter. There are no relevant variants, but again, we can do these cases in different steps, and I can save every step to the report basket. So all this will be saved. Every step can be saved for me to review later. When I come to the compound heterozygous filter, I see the same variants that I had seen before. To see the details, I just have to click, and the variant details window opens, and here I can go through all the information regarding the databases, also the nomad frequency information. I can go through all the bioinformatics scores here, and in this case also the splice, yeah, splice effect. And if I would like to just classify the variant, I can mouse over the criteria, and as I select the criteria, the classification, you can see that it's getting calculated, and I can also adjust the strength, and then the classification is calculated accordingly. I can add my comments and then confirm the classification. I can also choose to report the variant on a different transcript, and also add publications relevant for reporting from this view, which will appear in the report later. I can also choose to say that I will right away classify the variant from this view. So I can choose a classification, and then this will be now a classified variant. Now let's say I've completed the case and I would like to look at all the variants that have been classified. I simply have to come here and click it to see all the classified variants. Or I could also say, just show me all the variants that I have highlighted in the case that I want to review again. Now let's go into the report view. All the variants that have been classified and that I want to report are here. And now I can start entering all the details, like the disease. Then I can enter the variant category. So this would be my primary finding. I can enter the mode of inheritance. I can enter the genotype information and add comments if necessary, mark the case as solved. And I can also send it for a second level review or approve and complete the case if I'm confident of the case. Now the report will be available as a document or a PDF that you can upload to your LIMS system. But also everything in the report basket will be now available as a report attachment and attached to the case. And this can be reviewed at any time, at a later time as well. Now, there are several other features that I am not able to show you right now, but I would be happy to demonstrate all the different features on a one-on-one demo. So just get in touch with us and I will hand this over back.
Okay, let's go back to the slides. So we've covered this part of the workflow now, and would like to show you briefly how this will look like with CNVs, because this is the part that we have not covered now with in the demo. And this is just a screenshot how this view would look like. So this kind of table already looks familiar. We have it in all the views, and it looks just what Irene showed you before. So every row here is a variant detected. You have filter buttons that you could use, and you can also save all your findings in the report basket. You can review data on 2 different levels. So the CNVs detected that comprise maybe many different exons or even many different genes. Or you can review the data, really the raw data of the CNV analysis for every target region that is considered in the analysis. On top, we have a graphical representation of the data. So we have the chromosome here, quality metrics on top. You can review individual chromosomes or jump to specific variants by clicking in the grid, and then you would see exactly this variant also in the graphical representation. Here you see the distribution of the target region log2 values. So the green line basically corresponds to the normal state of 2 copies, and deletion would then look like this, that all the target regions here would be located below the threshold that is indicated with a red line here, and the algorithm automatically detects this as one big copy number variant, and this is indicated with this red bar here in the software. We've integrated DGV and decipher annotations so that you can have possibilities to find the disease-causing variants here. Again, you can filter with the press of a button in the same way that you do it in the variant grid, classify the variants, and this is available for small panels up to whole genomes. And this is just a screenshot. If you would like to see a clinical example where we can really go through all the details of the view, we can do this in a detailed software demo. There's no problem. So the variants that have been reported at the end, they are transferred to your in-house or local database in varvis, and this information can then be used to classify the next analysis. So if I classify a variant today as pathogenic, and tomorrow in the analysis, I will see that this has been classified as pathogenic before, and I can use this information in the next case. Also, annotations from external data sources are maintained automatically, and this also gives a very nice feature, the annotation update feature. So this is the view that also Irene showed you in the demo. We have a patient, an SNV and a CNV analysis have been performed. This is a rather old case from 2019, and we looked at this data again in 2021. And let's say this case was still negative, and now I want to take another look at this case and see if there's a new information available that would provide the answer. I can click on this arrow symbol here, and then what opens is the annotation update window. So you have an overview of all the annotation sources that come with varvis, and you see here in this column that version of the sources that has been used for the last annotation when I looked at it in 2021. And now, if I decide I would like to put this old analysis into a new look, I can press update annotations, and then the system basically annotates all the new information to the old case, and then I can maybe have the new findings in ClinVar or Alexis or literature will help to solve the old case finally. With Alexis, we also offer the possibility to share information within the whole network of varvis users, and this is done in an anonymized way. So let's say a patient A is analyzed in a laboratory X, and this for this patient, personal data has been entered, phenotype description, and all the list of variant is available, and the same thing is done for another patient in another lab. The information transferred to Alexis is then anonymized, so only the phenotype and variant information is shared together with like an anonymized ID. So if you now look at a new patient in your lab and enter all the information for your patient, and you see the list of variants, you would see for every variant how often it has been detected in the whole network. So in this case, we have 2 hits. You would see the 2 different phenotypes that have been entered for the patient, and you would see that these are 2 different families and 2 different labs that worked on the cases. And so what you would see is basically the allele frequency, the significance, all the associated HBO terms, but you have no access to patient identifiers. So this is privacy by design, but a very powerful tool also to find variants that have been classified only once in the past, and then this could maybe be the one extra information that you need to conclude in your case. So within varvis, basically you collect data on different levels. So for every individual patient, there's already comprehensive data capture. You see all the variants, all the artifacts, sample-specific metadata. While reviewing patients, you build up your own in-house local database. These are typically homogeneous data sets. You have full access to the analysis history, so you can review all cases at any time and use all the information aggregated in your local database. And then one level on top is then the data sharing network Alexis, where you can share the data with other varvis users. And this is, of course, compliant with the EU regulations. So coming back to the standardized workflow, we basically we provide we will provide the software for you, but we also basically outsource the bioinformatics part. You don't have to worry about this one. It's grayed out, and also the annotation management and the database management is also updated automatically. So there's also no need to worry about this. So you can fully concentrate on your patients and on classifying your variants. Apart from, on top of offering the software, we offer support and learning training tools. We regularly host webinars like the one today. There are blog articles available on the website, also mentioned from Ben before. And we are hosting a very nice training platform, the varvis Academy, where there are hands-on courses where you can, everything is explained that you need to know when you start using the software, like how to configure filter buttons, how to configure new users, how to solve exome cases with a hands-on training course for whole exome cases. Also, like basic information about carrier screening, for example. So there are many nice courses available. And yeah, if you would like to learn more, we offer an onboarding program that basically consists of 2 different phases. The first one is the testing phase. It typically starts with a demo presentation comparable to what we're doing today, but more interactive, hopefully. So we aim at answering all questions. Also, depending on the use case you're using, we will try to find a nice case example we will go through together. You can then use a 14 days free trial of the software where you can upload own analysis. And then you have 2 weeks time to test the software, try and test all the training material as well. We start typically with like an onboarding training beforehand, also with Q&A sessions, so that we make sure that you have all the information you know to decide if this is some tool that would work for you or not. If you would then like to take the next steps, we can set you up for clinical routine. Also, we have a program for this which includes software configuration, feedback regarding your QC parameters, the validation, technical support, and access to the varvis Academy. This is a question we usually get, and this is why we're answering it straight away. So what do I do with my knowledge base I have somewhere in another tool? What can I take this with me? And the answer is yes, we offer an import of existing data. So your current database would be, if you provide this as a VCF with the defined information you would like to import, for example, classification and comments, you would see this as an internal annotation source in varvis, so you will not lose out on information that you have gathered in the past. And this brings me to my final slide. So this is basically the feedback we received from customers, why they are happy using varvis. And some say they really like the QC feedback they got, and they were able to optimize the lab processes. They enjoy the the advantages of using a cloud-native CE medical device software. Another plus is the no investment in IT infrastructure, so ready to go in no time, basically. Equal performance and support across the globe. The support with our expert team is also a very big plus. And the cost savings through automated CNV analysis, and it scales nicely from small panels to even whole genome. And for this, I would like to hand over back to Jimena, and let's see if there are any questions.
Thank you very much, Yvonne. We indeed have some questions. Maybe I can start with one for Irene, maybe. It says, am I able to check a new gene in all patients to see if any of our patients carry variants in this gene?
Yes, we have a search feature where users are able to search for a given gene or also a variant. So you would be able to search given specific details, and you would have results and the patients who are carrying them. This is not a problem.
Maybe this can be answered by Yvonne. Hello, do you provide software demo that customers could test the software using their own data?
Yes, we do offer a software trial, free software demos, free software trial. I guess this question came in before we showed the slide. So yeah, no problem. Just get in touch and we're happy to show you in detail.
Yeah, we have another question for this part of the session. Can we change or define our own default transcript?
Yes. So the user can define the default transcript as we set up the pipeline, but also there is a whole gene management system where you will be able to set your own default transcript on which the variant must be called. But all other transcripts are also available in the variant view like I showed. So yes.
And we have an extra question for the first part. So maybe Ben, you can answer this one. How does your platform test report help me with the IVDR compliance of my own in-house test?
So this test report that we provide basically should be part of your own documentation. So if you would not be using our platform test, you would have to generate this data on your own. We already have a standardized procedure for that, and it has proven to be very helpful. And it also kind of this test report has also been reviewed by the notified body. So you can be sure this is a validated test method. So it saves you time.
Fantastic. Thank you all. And I would like just to, as a final remark, if anyone has further questions or want like a one-to-one demo or directly start a free trial or just get more general information, you can visit our webpage that you are looking at this address in this slide, or you can contact us at info@varvis.com. And well, thank you to all the participants, to the speakers, and I hope you have enjoyed as much as I did. Thank you. Goodbye.